Hi there! In this video tutorial I'll show you how to use the GeneProf Web Services to load some data from GeneProf directly into an active RNA-seq. I'll be working with this experiment as an example in which Kirigan and his colleagues have depleted microRNA biogenesis in two different cell populations, and they have generated corresponding control datasets. The data has already been analyzed in GeneProf and is available in this dataset, which combines all the different experimental samples and has also calculated logarithmic fold changes and p-values for those. Now I wanted to use the GeneProf Web Services to load this data into an active R session and then create some plots. GeneProf Web Services provide a convenient interface for external applications to access GeneProf data, and there are a number of tutorials available online that show you how to use it. Now for this example, we just want to use the GeneProf web service called data as binary files. Now this is the documentation that details how to use this web service. And I'll just open my R session here. What I first want to do is to load the rcurl library, which is what we'll be using to open a connection to the GeneProf web service. So I say library rcurl and then open a URL connection where I use the URL of this web service, replacing the ID placeholder with the identifier of the dataset that I want to load in. So I just copy and paste it over here and then load this URL connection. Oops, there was a little typo, so be very careful about what you type in. Just correct that, say load URL collection, close the URL connection and remove the object from the workspace to avoid cluttering. And I see my workspace now contains an object called GeneProf data. This is the dataset that I've loaded in. It contains the same columns that you see in the background of the GeneProf interface. And I can do with that whatever I want to do in R. For the purpose of this tutorial, I'll first plot a scatterplot panel for the RPKM values in the dataset. So here we have the four datasets plotted against each other. We see that overall they correspond quite well. Most genes don't change very much, but there's a set of genes that changes between the different conditions. Now we can highlight differentially expressed genes in this plot as well, and then highlight those that are consistently differentially expressed in both different cell types upon abrogation of microRNA biogenesis. There are 118 of those. I'll just replot the scatterplot panel, making those differentially expressed genes red and leaving the rest gray. Of course, I can also look up the names of the genes or any of the other annotations available in the dataset. You see there are other examples available for data that I could load from GeneProf. There's a whole number of tutorials available, amongst others one for R, that shows you how to load public gene expression data very quickly into an active R session and how to exploit that from within. So I hope you enjoyed this short tutorial and see you next time.